membrane potential, looking at the nurse potential for sodium and the nurse potential for potassium. What I mean by nurse potential is what is the electrical difference across the cell membrane that stops the flow of ions down their concentration gradient, right? That's the nurse potential. All right, well, we don't have theoretical cells, we have real cells. So if we look at how this plays out when everything is in motion, okay, so here is our potassium channel, right? So we can see potassium leaving the cell down its concentration gradient. Here's our sodium channel. Sodium's coming in from the outside down its concentration gradient. Okay, so then we have our sodium-potassium exchange pump, which we talked a little bit about yesterday as well. That is pumping potassium in 2 at a time and sodium out 3 at a time using ATP. So when we have all of these different forces at work at the same time, we end up with our measured membrane potential. Okay? Now, the measured membrane potential in this case is minus 70 millivolts. That number is, it varies according to which cell you're looking at in the body. So like if you were to measure the membrane potential of an epithelial skin cell, it's about minus 70. Many of the cells that we talk about, like neurons, are going to have a lower resting membrane potential than that, so like minus 90. And like skeletal muscle cells, depending on their degree of excitement, they can have a membrane potential as high as minus 50 or as low as minus 80. So the numbers don't matter. Like, I'm not going to ask you what is the resting membrane potential of X cell, because the truth is it's a variable, right? It's not the specifics don't matter. It's more, okay, minus 70 is somewhere between 50 and 100, right? And it's minus on the inside. So the detail here, like the number, is less important than the what it means. All right, so one of the things it means is, you know, we said that the nurse potential for sodium and potassium are sort of opposite. Plus 61 for sodium, minus 94 for potassium. Okay, well, if we measure the membrane potential, we get minus 86. All right, so what does that mean? It means that if we open a sodium or potassium channel, those molecules are going to move because neither are at their nurse potential, right? Neither are at equilibrium. So if we, regardless of what the membrane potential is, if we open a channel, because it's not perfectly balanced, those molecules are going to move and the membrane potential is going to change, right? So what this diagram is showing you is, first off, what are the relative concentrations inside and outside, right? So sodium high outside, sodium low inside. Potassium reversed, high inside, low outside. Then we have the nurse potential. So for sodium, that's about plus 61. For potassium, that's about minus 94. If we combine these two in the same cell and we add our leak channels, that brings us to about minus 86, okay? Then if we add the effect of the sodium-potassium pump, that brings us to about minus 90. So that's how we get to minus 90 for a neuron, is we combine the nurse potential for sodium and potassium, and then we add the sodium-potassium pump. Why does the pump matter? Because remember that the pump, come on, go backwards. There we go. The pump is, in a sense, working opposite the leak channels all the time. Do you see that? So sodium leaks in and is pumped out. Potassium leaks out and is pumped in. See that? Now, you could argue, like, one of the things I like you to think about sometimes is if you were going to redesign the human being, what improvements would you make, right? Well, one of the things you might improve is if we got rid of the leak channels, our sodium-potassium pumps would have much less work to do, right? So it would actually make us quite a bit more efficient in terms of our energy metabolism, but that isn't what we have. We're leaky to sodium and potassium. And our body kind of uses that leak